A bare foot descends through warm, waist-deep water and meets what feels like a patch of coral. In the next heartbeat, the reef explodes into agony. The pain does not rise gradually. It detonates, radiating up the limb as if the seabed itself has grown fangs. What triggered it was not a chase, not a strike in open water, but a compression event measured in millimeters. Beneath that patch of rubble, a living organism converted weight into venom, transforming downward pressure into biochemical retaliation. The stonefish does not pursue its enemy. It waits for gravity to deliver one. On the reef floor, where sunlight fractures into moving mosaics and shadows break up every outline, the stonefish abandons the architecture of a typical fish. Its body is squat, irregular, and asymmetrical, with skin textured into lumpy protrusions that mimic encrusting algae, dead coral heads, and limestone fragments. Chromatophores in its dermis adjust pigmentation to match surrounding substrates, while filamentous appendages disrupt any remaining silhouette. This is not simple camouflage, it is morphological surrender. The animal erases its identity to become part of the substrate itself. Predators scanning for bilateral symmetry and reflective scales see nothing. Prey drifting over the reef interpret the shape as geology. The stonefish survives by collapsing the distinction between organism and environment. Yet the camouflage is only the first layer of strategy. Rising from the dorsal surface are stout spines connected to venom glands embedded at their base. Each spine is sheathed in integument that tears under sufficient pressure, allowing venom to flow along grooves and into the compressing tissue. The delivery mechanism is passive but devastatingly efficient. The more force applied, the deeper the spine penetrates and the more venom is expressed. This is a system designed not for aggression, but for consequence. The reef becomes a biochemical perimeter, and the stonefish defends territory without movement. The venom itself is a complex mixture of proteins that disrupt cellular membranes, alter neuromuscular transmission, and induce intense inflammatory cascades. Upon envenomation, pain fibers fire relentlessly as toxins trigger local tissue damage and systemic responses. Cardiovascular effects may follow, as certain components interfere with cardiac muscle contraction and vascular tone. The result is not merely deterrence but incapacitation. A predator that attempts to swallow or bite carelessly may recoil in shock, its nervous system overwhelmed before it can complete the act of predation. The stonefish does not roar or flash warning colors, it enforces respect through chemistry. But the ecological twist lies in the fact that the stonefish is not primarily a defensive organism. Beneath the venomous armament is an ambush predator of remarkable subtlety. When small fish or crustaceans drift within range, the stonefish employs a rapid suction feeding strike. Its jaws protrude, the buccal cavity expands, and a pressure differential draws prey inward in a fraction of a second. This feeding strategy relies on hydrodynamic manipulation rather than speed across distance. By remaining motionless and blending into the benthos, the stonefish allows prey to approach voluntarily. The reef's productivity, fueled by nutrient upwelling and symbiotic coral metabolism, ensures a steady supply of unsuspecting victims. The venom that terrifies larger animals is rarely used in hunting. Instead, stealth and negative pressure do the work. The duality is striking. What appears to be a sedentary defensive relic is in fact a specialized predator exploiting the reef's visual noise. Coral reefs are among the most structurally complex ecosystems on the planet, their three-dimensional architecture creating countless microhabitats and occluded sightlines. Visual predators must constantly filter irrelevant textures from meaningful motion. The stonefish exploits this sensory overload by becoming part of the background algorithm. It thrives, not despite the reef's chaos, but because of it. Energy economics further explain its strategy. Maintaining constant locomotion in warm, oxygen-variable shallows would demand high metabolic output. By remaining still for extended periods, the stonefish minimizes caloric expenditure while maximizing ambush efficiency. Its musculature is optimized for explosive bursts rather than sustained swimming. In a habitat where competition for prey can be intense and predators abound above and below, reducing visibility reduces conflict. Camouflage lowers encounter rates. Venom discourages escalation. The combination forms a layered defense that conserves energy while preserving lethal capacity. The narrative reversal deepens when one considers that the stonefish's immobility is not weakness, but calculated adaptation. Many reef predators advertise their toxicity with bold coloration, relying on aposematism to deter attack. The stonefish does the opposite. It hides its warning until contact occurs. This concealment might seem risky, yet it reduces harassment from species capable of recognizing warning patterns but willing to test them. By blending in, the stonefish avoids drawing attention altogether. Only when misidentification occurs does the venom assert ownership of space. The reef floor becomes a silent boundary line. However, even this formidable strategy exists within a web of vulnerability. Larger predators, such as certain sharks and rays, may consume stonefish with relative immunity, their thick dermal layers and behavioral tactics mitigating envenomation risk. Human intervention adds another layer of threat, as habitat degradation and coral bleaching alter the structural complexity upon which the stonefish depends. When reefs flatten due to thermal stress and acidification, the mosaic of textures that once concealed the stonefish becomes simplified. Camouflage loses fidelity in uniform rubble fields. The predator that mastered disguise may find itself exposed by environmental change. There is also an evolutionary compromise embedded in its design. The same dorsal spines that defend against trampling can limit flexibility and speed. The body plan optimized for benthic ambush is poorly suited to open water escape. If flushed from cover, the stonefish cannot outswim agile predators for long. It relies on stillness as both shield and sword. The venom compensates for physical limitations, transforming a slow body into a hazardous obstacle. In this way, what appears to be morphological deficiency is reframed as specialization. Reproduction follows the reef's seasonal rhythms. During spawning events, eggs and sperm are released into the water column, entrained by currents that disperse genetic material across reef patches. Larval stages drift in planktonic suspension before settling onto suitable substrates, where metamorphosis aligns body shape with benthic life. This dispersal ensures genetic connectivity among fragmented reefs, allowing populations to persist despite localized disturbances. The stonefish is not isolated rock, but participant in a dynamic coastal network shaped by currents, tides, and larval recruitment. Even the venom has a broader ecological resonance. Envenomation events can alter predator behavior, teaching avoidance and reinforcing spatial memory. A single painful encounter may recalibrate how an animal navigates the reef, indirectly shaping movement patterns and territorial boundaries. In this sense, the stonefish exerts influence beyond direct predation. It modifies the behavioral landscape, adding invisible constraints to the physical terrain. As dusk settles and reef activity shifts toward nocturnal species, the stonefish remains anchored, a patient node in a system of flows. Plankton pulses with tidal cycles, fish schools reorganize, and crustaceans emerge from crevices. The stonefish does not announce its presence. It waits, skin indistinguishable from calcified debris, spines aligned like dormant thorns. The reef appears peaceful, yet beneath that calm lies a calculus of risk, written in venom glands and hydrodynamic physics. The final reversal is almost philosophical. The stonefish's strategy is not domination through overt force, but governance through consequence. It transforms error into injury, misjudgment into pain. The reef floor becomes a domain where perception determines survival. Those who read the textures correctly pass unharmed. Those who fail are reminded that the boundary between life and stone is porous. In the intricate theater of coral ecosystems, the stonefish stands as a testament to evolutionary ingenuity, proving that the most formidable weapons may be those that wait in silence, disguised as nothing at all, until the moment the world presses down and discovers that the ground itself can strike back. The reef tightens without a sound. A school of juvenile damselfish compresses against a coral head, their bodies flashing in synchronized microbursts of silver as something glides toward them with impossible calm. There is no surge of speed, no explosive lunge, only a slow expansion of striped fins, an ornate fan unfurling like a ceremonial banner. The lionfish does not rush. It rearranges the geometry of the water itself. In that widening circle of spines, escape routes vanish one by one, and within a heartbeat the school fractures. One fish hesitates. The predator's mouth opens, the buccal cavity flares, and suction replaces distance. Beauty has just inhaled a life. The lionfish appears ornamental, as though sculpted for admiration rather than survival. Its elongated dorsal spines rise in delicate arcs, each one sheathed in venom glands, capable of delivering a painful and physiologically disruptive sting. Its pectoral fins spread wide and translucent, striped in bold contrasts of cream and rust that should, by conventional logic, make concealment impossible. Yet the conspicuousness is not a mistake. It is an evolutionary declaration. The lionfish advertises its toxicity openly, converting visual contrast into deterrence. In ecosystems where predators learn quickly, high-contrast patterning functions as aposematic signaling, broadcasting chemical defense before contact ever occurs. The lionfish does not hide its weapon. It frames it. Venom in this case is not merely punitive but strategic. Each dorsal spine is connected to a venom gland that releases toxins when the spine penetrates tissue, typically during attempted predation or accidental contact. The venom contains proteins that disrupt neuromuscular signaling and trigger intense pain responses, often accompanied by localized swelling and systemic symptoms. For a predator contemplating a bite, the cost-benefit analysis shifts dramatically after a single encounter. Pain becomes memory, and memory shapes behavior. The lionfish leverages that learning curve. By surviving an initial confrontation, it earns a reputation that protects it thereafter. Chemical warfare becomes a long-term investment in safety. Yet the narrative reversal emerges when we consider that the lionfish is not simply a defensive icon. It is an extraordinarily efficient predator in its own right. Unlike ambush specialists that rely on invisibility, the lionfish manipulates perception through motion and structure. Its wide pectoral fins are not decorative excess, they are tools of containment. By slowly herding prey against reef surfaces or into corners of branching coral, it reduces the angular freedom of escape. The fins create a visual and physical barrier, a moving wall that narrows the prey's decision space. In the confined geometry of a reef, even a modest constraint can tip survival odds. When the moment arrives, the lionfish employs rapid suction feeding, expanding its mouth cavity to generate negative pressure that pulls prey inward in a fraction of a second. The apparent slowness conceals a strike powered by hydrodynamic precision. The reef ecosystem magnifies this strategy. Coral reefs are complex labyrinths of crevices, overhangs, and vertical faces, where small fish depend on quick directional changes to evade predators. The lionfish exploits this reliance on maneuverability. By positioning itself deliberately and extending its fins to block potential escape vectors, it transforms agility into vulnerability. The prey's nervous system, tuned to react to high-speed pursuit, is less prepared for gradual encirclement. The lionfish hunts not by outrunning, but by outmaneuvering in slow motion. Its elegance is tactical. Energy efficiency underlies this method. Sustained high-speed chases demand elevated metabolic output, particularly in warm tropical waters where oxygen solubility is limited and aerobic capacity can become a constraint. The lionfish circumvents this limitation by minimizing movement until the final instant. It hovers using subtle fin adjustments, conserving energy while assessing position. When it strikes, the expenditure is brief and decisive. Over time, this strategy yields a favorable balance between caloric intake and metabolic cost. Venom reduces predation risk, and controlled hunting conserves resources. The lionfish embodies a convergence of chemical defense and mechanical economy. In its native Indo-Pacific range, this balance integrates into a web of co-evolved relationships. Predators that recognize the lionfish's warning pattern avoid it, while prey species adapt to its hunting style through vigilance and habitat choice. The system stabilizes through mutual pressures. But when transported beyond its evolutionary context, as in parts of the Western Atlantic and Caribbean, the lionfish's conspicuous display loses its learned meaning. Native predators unfamiliar with its aposematic signals may hesitate too late or avoid it entirely after painful encounters, reducing natural control. Meanwhile, prey species unaccustomed to its herding behavior fail to recognize the gradual encirclement. The result is a predator whose efficiency becomes amplified by ecological naivete. This is where the ecological twist deepens. The same traits that evolved for balance within one ecosystem can destabilize another when introduced without checks. Lionfish populations can expand rapidly, consuming juvenile reef fish at rates that alter community composition. By reducing populations of herbivorous fish that graze on algae, lionfish indirectly influence coral health. Algal overgrowth can suppress coral recruitment, shifting the reef's structural complexity over time. A single predator, armored in stripes and venom, becomes a catalyst for trophic cascades. Its beauty masks systemic consequence. Yet even here, the lionfish's dominance is not absolute. Certain large groupers and sharks have been observed preying upon lionfish, sometimes learning to flip them or approach from angles that avoid the spines. Human divers, equipped with protective gear and targeted removal efforts, also become a controlling force. The lionfish's conspicuousness, once a shield, becomes a liability when predators adapt. Its warning display makes it visible not only to avoiders, but to hunters who have learned to neutralize its defense. The advertisement cuts both ways. The venom itself illustrates another paradox. It is potent enough to deter many threats, yet it is not optimized for active predation. Unlike venomous snakes that inject toxins to immobilize prey, the lionfish relies primarily on mechanical capture. The venom is insurance, not primary weaponry. This division of labor allows the lionfish to specialize its hunting apparatus without sacrificing defense. Its spines enforce personal space. Its mouth secures sustenance. Chemistry and biomechanics operate in parallel, each enhancing the other's effectiveness. As twilight descends over the reef, lionfish often become more active. Many small reef fish reduce vigilance under low light, relying on shelter and camouflage. The lionfish's striped pattern, already disruptive in bright conditions, becomes even harder to pass in shifting shadows. Its slow advance appears almost suspended in time, as though it floats outside the urgency of the reef. But within that stillness lies acute sensory processing. Vision tuned to contrast and movement allows it to detect subtle shifts in prey position. The reef pulses with nocturnal transitions, and the lionfish exploits those rhythms with patient intent. The final reversal lies in the perception of ornamentation itself. To the human eye, the lionfish resembles an exotic display, an aquarium centerpiece framed by flowing fins. That aesthetic appeal has facilitated its spread through the ornamental fish trade, a pathway that inadvertently reshaped entire ecosystems. The trait that invites admiration also enabled dispersal. Elegance became vector. In this way, the lionfish demonstrates how evolutionary signals can intersect with human behavior, creating outcomes far beyond their original ecological function. Hovering among coral branches, spines erect yet motionless, the lionfish commands space without haste. Its stripes pulse faintly in refracted light, a warning written in contrast and symmetry. Prey gather, unaware that geometry is shifting around them. Predators pass at a distance, remembering pain encoded in nerve endings. The reef continues its ceaseless exchange of energy and competition, and within it, the lionfish occupies a niche forged by chemistry and design. It is not speed that grants dominance, nor brute strength. It is the seamless fusion of display and deterrence, of slow motion and sudden force. In the theater of coral ecosystems, the lionfish proves that venom can be worn like regalia. That advertisement can be armor, and that elegance, when engineered by evolution, is often the most deceptive weapon of all. The fish does not see the strike. One instant it is threading through a maze of branching coral, muscles flickering in rhythmic bursts powered by aerobic precision, and the next its body stiffens as though an invisible current has surged through its spine. The sea snake has already withdrawn, jaws closing with minimal ceremony, leaving behind only a pair of punctures so small they barely disturb the scales. There is no thrashing pursuit, no drawn-out struggle clouding the water with sediment. Within moments, neuromuscular control begins to falter. The prey's own physiology betrays it as venom molecules diffuse through muscle tissue, binding to receptors essential for contraction. The ocean has delivered a verdict with surgical efficiency. Sea snakes carry the silhouette of terrestrial ancestry into a world that reshapes every constraint. Their bodies remain unmistakably serpentine, elongated and limbless. But evolution has re-engineered them for hydrodynamic performance. The tail flattens into a paddle, increasing surface area for propulsion, while valved nostrils sit atop the snout, allowing quick breaths at the surface before submergence. Internally, a single elongated lung runs much of the body length, functioning not only in respiration but in buoyancy control, its air volume adjusted to hover neutrally within the water column. The transformation is partial yet profound. They are reptiles that never fully surrendered to the sea, yet have become inseparable from it. Venom is their central innovation, and in marine ecosystems it serves a different calculus than on land. In terrestrial ambush, a snake may track envenomated prey over distance, following scent trails until toxins take effect. In the ocean, such luxury rarely exists. A wounded fish can vanish into labyrinthine coral within seconds, oxygen debt mounting with every moment of pursuit. For a reptile dependent on lungfuls of atmospheric air, extended chases are metabolically costly. The sea snake resolves this dilemma through potency. Its venom, often rich in neurotoxins and myotoxins, targets the neuromuscular junction, disrupting the transmission of signals between nerve and muscle. Paralysis follows swiftly, reducing the window in which prey can escape. Chemistry compensates for limited oxygen reserves. This efficiency reshapes predator-prey dynamics. By immobilizing fish rapidly, sea snakes minimize energy expenditure and reduce exposure to their own predators. Reef sharks, large groupers, and birds of prey may exploit vulnerability near the surface. A drawn-out hunt would advertise weakness. Instead, the sea snake glides with deceptive serenity, body undulating in lateral waves that generate forward thrust without turbulence. Its motion is almost hypnotic, a visual calm that belies biochemical lethality. In a habitat where movement often signals threat, its quiet advance is camouflage of behavior. The narrative reversal emerges when we consider that this fearsome predator is physiologically fragile in ways most observers overlook. As reptiles, sea snakes must surface to breathe. Their dives are constrained by lung capacity and metabolic rate, forcing a rhythm of descent and ascent that intersects with tides and prey activity. Unlike fish with gills extracting oxygen continuously, the sea snake lives by intervals. Yet this limitation becomes an adaptive advantage. By controlling buoyancy through lung inflation, it can hover with minimal muscular effort, conserving energy between strikes. The same lung that limits dive duration grants fine control over vertical position. Weakness becomes instrument. Their skin, too, reflects compromise. Salt water poses osmotic challenges, drawing water out of tissues and flooding cells with ions. Sea snakes possess specialized sublingual salt glands that excrete excess sodium and chloride, maintaining internal balance despite immersion. This adaptation allows them to drink seawater without succumbing to dehydration, a capability rare among reptiles. Even their scales are smoother than those of terrestrial relatives, reducing drag as they navigate coral channels. Each anatomical adjustment represents incremental negotiation with marine physics. Reproduction introduces another layer of divergence. Many sea snake species are viviparous, giving birth to fully formed young in the water, rather than returning to land to lay eggs. This reproductive shift anchors them to the sea entirely, severing the final tether to terrestrial origin. The young emerge as miniature predators, already equipped with venom and instinct. The ocean becomes cradle as well as battlefield. The lineage has crossed a threshold where land is no longer sanctuary but relic. Their venom, potent as it is tailored primarily for fish physiology. Human encounters, though capable of severe consequence, are rarely the intended target. The toxins act by binding to specific molecular receptors, and while these receptors exist across vertebrates, evolutionary fine-tuning favors piscine prey. This specificity underscores a broader truth. Venom is not indiscriminate malice, but biochemical precision honed by selective pressure. The sea snake's arsenal reflects millions of encounters with slippery, fast-moving fish rather than with terrestrial intruders. Within reef ecosystems, sea snakes occupy a niche that intersects with eel burrows and crevice-dwelling fish. Their slender bodies can probe cavities inaccessible to bulkier predators. By exploiting these refuges, they influence the distribution and behavior of species that might otherwise dominate microhabitats. This subtle predation can cascade through trophic levels, altering competitive balances among small reef fish. The sea snake is not merely a solitary hunter, but a participant in community regulation. Yet they are not invulnerable. Larger sharks may consume them, immune or resistant to their venom. Some fish have been observed harassing sea snakes, nipping at tails to drive them from territory. And human fishing practices pose significant risk, with sea snakes often entangled in nets as bycatch. Their air-breathing nature becomes perilous when trapped underwater. The predator that immobilizes prey through oxygen-efficient venom can itself succumb to suffocation in an indiscriminate mesh. The hierarchy of threat shifts with context. As dusk approaches, plankton migrate upward in vertical tides, and small fish follow, creating transient feeding corridors. Sea snakes adjust accordingly, timing surface intervals with prey density. Their sensory systems detect subtle movements in the water column, lateral undulations translating into three-dimensional awareness. Vision, chemoreception, and mechanoreception integrate into a fluid hunting algorithm. The ocean at twilight is a theater of gradients rather than boundaries, and the sea snake moves within those gradients with calibrated restraint. The final reversal lies in perception. To human observers, the serpentine form evokes ancient terrestrial fears, an instinctive unease carried into the sea. Yet the sea snake is less aggressor than adapter, less terror than testament to evolutionary possibility. It represents a lineage that re-entered the ocean and solved its constraints through chemistry and design. The calm exterior is not deception but equilibrium, a balance between oxygen limitation and venom efficiency, between ancestral inheritance and marine innovation. In the dimming blue of evening water, a sea snake surfaces briefly, nostrils breaking tension before slipping beneath again. It leaves no wake of drama, no violent splash. Somewhere within the reef, a fish stiffens and sinks, muscles silenced by molecular interference. The predator returns to claim it, conserving energy for the next measured glide. In this exchange, the ocean reveals another of its paradoxes, that lethal capacity can travel in silence, that elegance can mask efficiency, and that the most decisive forces in marine ecosystems often operate not through spectacle, but through the invisible choreography of venom and breath. The strike collapses in mid-water. A reef predator lunges, jaws flaring, and instead of closing around sleek muscle, its mouth meets a sphere that did not exist a heartbeat earlier. The target has transformed. Water floods into a highly elastic stomach through rapid buccal pumping, and the fish's body expands into a rigid globe studded with spines that were once flat against the skin. The predator recoils, teeth scraping against an architecture designed to resist swallowing. In the instant between intention and ingestion, the pufferfish has rewritten the physics of the encounter. What was bite-sized has become impossible. Inflation is not a trick of air, but of hydraulics. Most marine puffers are physiologically incapable of holding large volumes of air underwater. Instead, they force water into a specialized, distensible stomach lined with folds that can stretch several times beyond resting capacity. The ribs are reduced or modified, allowing the body cavity to expand without structural fracture. Muscles coordinate the intake in rapid cycles, and the gill openings constrict to prevent backflow. This is not panic. It is a reflex pathway honed by selection triggered by sudden threat cues processed through sensory circuits attuned to looming motion. Within moments, the puffer's silhouette shifts from streamlined to spherical, and the predator's gape limitation becomes the puffer's shield. Spines intensify the geometry. In many species, dermal spines lie embedded and flattened against the body during normal swimming, minimizing drag and preserving hydrodynamic efficiency. Upon inflation, these spines pivot outward, transforming smooth skin into a bristling surface. The combination of increased diameter and rigid protrusions complicates the mechanics of ingestion. Predators that rely on suction feeding encounter a volume too large to draw in. Those that bite face a mouthful of resistance that risks injury. The pufferfish does not flee at high speed. It alters the equation of capture. Yet the deeper defense lies beneath the skin. Many pufferfish harbor tetrodotoxin within their tissues, a potent neurotoxin that blocks voltage-gated sodium channels essential for nerve impulse transmission. When these channels are inhibited, action potentials cannot propagate along nerves, and muscles fail to contract. The toxin does not cause pain in the dramatic way of venomous stings. It causes silence. Paralysis emerges as a systemic consequence of molecular interference. The pufferfish does not inject this toxin into attackers. It embeds it within its own flesh, turning consumption into hazard. The origin of tetrodotoxin reveals a narrative twist that expands the story beyond a single organism. Puffers do not synthesize the toxin entirely on their own. In many cases, symbiotic or dietary bacteria produce the compound, which then accumulates in the fish's liver, ovaries, and skin. The puffer becomes a reservoir rather than a manufacturer, integrating microbial metabolism into its defense strategy. The fish that appears solitary and self-contained is in fact part of a biochemical network that spans microorganisms and marine food webs. Poison is not just a property of tissue, it is a collaboration. This integration reshapes predator behavior at an ecological scale. A predator that survives an attempted meal of a toxic puffer may learn avoidance, adjusting its foraging patterns to exclude similar shapes. Through this feedback loop, the puffer's defense influences the structure of predator diets and indirectly affects competition among other prey species. If a predator reduces consumption of pufferfish, it may increase pressure on alternative prey, altering community dynamics. The toxin thus radiates outward through trophic pathways, shaping the reef beyond the immediate encounter. Inflation itself carries cost. Expanding the body cavity requires muscular effort and temporarily compromises mobility. While inflated, the puffer sacrifices speed and maneuverability. The spherical form, though resistant to swallowing, is poorly suited to sustained swimming. In strong currents, an inflated puffer becomes vulnerable to displacement. The defense is potent but energetically expensive, a short-term strategy rather than a permanent state. Once the threat subsides, the fish must expel water and return to its streamlined shape to resume efficient locomotion and foraging. Survival depends on timing as much as on toxin. Within reef ecosystems, pufferfish occupy varied niches. Some graze on algae, others crush hard-shelled invertebrates with powerful beak-like teeth formed from fused dental plates. These teeth grow continuously, worn down by grinding coral rubble and mollusk shells. By feeding on organisms that bore into coral or overgrow substrates, puffers participate in the reef's balance between construction and erosion. Their diet can influence algal cover, indirectly affecting coral recruitment and competition. The fish that transforms into a spiny balloon also contributes to the reef's metabolic exchanges. The narrative reversal deepens when considering that the toxin, while lethal in sufficient quantity, is tolerated by the puffer itself. Its own sodium channels possess structural differences that confer resistance to tetrodotoxin's binding. What is catastrophic for predators is neutral within its host. This molecular adaptation underscores a broader principle of evolutionary arms races. Defense and self-immunity must evolve in tandem. The puffer's tissues become a biochemical fortress, not only because they contain poison, but because they are impervious to it. Human culture intersects with this biology in striking ways. In certain regions, pufferfish are prepared as delicacies, their toxic organs carefully removed by trained hands. The margin for error is narrow. Residual toxin can induce paralysis and respiratory failure. This relationship between danger and desire mirrors the ecological dynamic between predator and prey. The puffer's flesh becomes both temptation and test, reinforcing the idea that toxicity is a boundary that can be respected or breached at great cost. The double barrier of inflation and poison represents a layered defense rarely matched in marine life. First, the predator must overcome mechanical impossibility. If it succeeds in swallowing, it confronts chemical peril. This redundancy reduces reliance on a single mechanism. Even if spines fail to deter, toxin may dissuade future attempts. The pufferfish embodies the principle that survival can be engineered through overlapping safeguards rather than speed or stealth. Yet puffers are not invulnerable. Certain sharks and large fish have developed tolerance or strategies to handle them safely, sometimes biting around spines or avoiding the most toxic tissues. The evolutionary contest persists. Defense invites countermeasure. Over time, selective pressures sculpt both sides of the interaction, refining toxins, altering behaviors, and adjusting thresholds of risk. The puffer's absurd silhouette, inflated and bristling, is one moment in a continuous negotiation between prey and predator. As dusk settles and reef activity shifts, puffers retreat into crevices, reducing exposure to nocturnal hunters. Their ability to wedge into tight spaces, inflate slightly to anchor themselves, and resist extraction provides additional security. Even in rest, their morphology serves defense. The reef is not merely backdrop, but partner, offering cavities that complement the fish's strategy. The final reversal lies in perception. To an observer, the inflated puffer may appear comical, a bloated caricature suspended in water. But that absurdity is a deliberate signal encoded in form and chemistry. It communicates that the reward of predation has collapsed, that the cost now outweighs the gain. The pufferfish does not chase or threaten. It redefines the predator's calculation. In the shifting blue of coastal seas, where countless species compete for survival, the pufferfish stands as an example of turning vulnerability into architecture. Its soft body becomes sphere, its tissue becomes toxin, its microbial allies become arsenal. Calmly grazing among coral heads, it seems unremarkable until challenged. Then, in a swift transformation, it announces that the boundary between prey and peril is thin and mutable. In the ocean's grand design, some creatures endure not by outrunning danger, but by making the act of consumption itself the greatest risk of all. The fish vanishes mid-fin beat. One moment it is drifting lazily between coral heads, drawn by the promise of shelter or stray plankton, and the next it is convulsing in a halo of translucent threads. The tentacles close with no visible muscles, no audible snap. Instead, the attack unfolds at the scale of cells. Thousands of microscopic capsules discharge in synchrony, each one firing a barbed filament that penetrates tissue and injects venom before the prey's nervous system can register the mistake. The sea anemone does not lunge. It waits for contact, and when contact comes, chemistry does the rest. Rooted to rock or reef by a muscular pedal disc that secretes adhesive compounds, the sea anemone appears less like an animal than like ornamental flora. Its column rises from the substrate, crowned by a ring of tentacles that sway with tidal rhythm. But this stillness is not surrender. It is strategic positioning in a current that carries opportunity. In coastal waters, plankton, larvae, and small fish move with oscillating flows driven by tides and wave action. The anemone extends its tentacles into this conveyor belt, transforming the motion of water into feeding probability. Each tentacle is lined with cnidocytes, specialized cells that house nematocysts primed to fire upon mechanical or chemical stimulation. The discharge is among the fastest biological events known, a micro-explosion of osmotic pressure that propels the filament outward with extraordinary acceleration. Venom delivered through these filaments is tailored for rapid immobilization. Neurotoxic peptides disrupt ion channels in prey cells, interfering with nerve signaling and muscle contraction. Paralysis follows quickly, reducing thrashing that might otherwise tear delicate tissues. Once subdued, the prey is guided toward the central mouth, where digestive enzymes begin extracellular breakdown before nutrients are absorbed into the gastrovascular cavity. The anemone's body plan is deceptively simple, a single opening serving both ingestion and waste expulsion, yet within that simplicity lies functional elegance. The tentacles serve as both trap and shield, feeding appendage and defensive perimeter. The ecological twist lies in the paradox of vulnerability. The sea anemone lacks the mobility of fish and the armored shell of crustaceans. It cannot flee a predator nor withdraw into rigid casing. Its softness appears liability. Yet the same softness permits flexibility and expansion, allowing tentacles to extend and retract with changing conditions. The anemone's reliance on venom compensates for physical fragility. Rather than investing energy in locomotion or skeletal reinforcement, it invests in microscopic weaponry and chemical deterrence. In doing so, it converts rootedness into advantage. Currents shape its life profoundly. Flow rate determines prey delivery, oxygen exchange, and waste removal. Too little current and food becomes scarce. Too much and tissue risks damage. Many anemones select microhabitats where hydrodynamic forces strike balance, anchoring to crevices that moderate turbulence while maintaining access to nutrient-rich streams. In these locations, the anemone becomes a node in the reef's metabolic network. Dissolved oxygen diffuses across its surface, carbon dioxide exits, and nitrogenous waste enters surrounding water to be processed by microbes and algae. Even in stillness, it participates in biochemical exchange. Some sea anemones form symbiotic relationships with photosynthetic algae, known as zooxanthellae. These microscopic partners reside within the anemone's tissues, converting sunlight into sugars through photosynthesis. In return, the anemone provides shelter and access to carbon dioxide and waste nutrients. This mutualism blurs the boundary between predator and producer. By day, the anemone harvests energy from light via its symbionts. By night, it captures prey through venomous tentacles. The organism that appears purely carnivorous is in fact partly autotrophic. Its identity shifts with the sun. Perhaps the most celebrated partnership is with clownfish, which navigate the anemone's tentacles unharmed. Through a process of acclimation and mucus coating that masks triggering cues, clownfish avoid nematocyst discharge and gain refuge among the stinging arms. The anemone benefits in turn from increased water circulation as the fish dart among tentacles, improving oxygenation and waste removal. The fish may also lure prey or deter predators that might graze on tentacles. What first appeared to be solitary reef furniture reveals itself as host to dynamic alliance. The anemone is not isolated but embedded in symbiotic web. This relationship introduces another reversal. The tentacles that incapacitate one fish provide sanctuary to another. The distinction lies in biochemical recognition. Nematocysts are triggered by specific mechanical and chemical signals. Clownfish adapt by adjusting their surface chemistry to avoid stimulation. The anemone's defense system is selective, not indiscriminate. It distinguishes between ally and intruder at the molecular level, underscoring the precision of its weaponry. Reproduction further complicates the narrative. Many sea anemones can reproduce both sexually and asexually. In sexual reproduction, gametes are released into the water column, where fertilization yields planktonic larvae that disperse before settling onto new substrates. Asexual reproduction through budding or fragmentation allows clones to establish colonies. A single anemone may thus represent both individual and collective, expanding its genetic presence across reef surfaces. The rooted predator multiplies without movement, extending its influence spatially through division. Predators of sea anemones exist despite their defenses. Certain nudibranchs and butterfly fish have evolved tolerance to their venom, feeding selectively on tentacles. These interactions reflect ongoing evolutionary negotiation, where toxin potency and resistance co-evolve. The anemone's chemical arsenal deters many but not all. Its survival depends on balance between deterrence and vulnerability. As tides shift and light angles change, the tentacles respond subtly. During low tide exposure in shallow pools, some anemones contract into compact forms to reduce desiccation risk, retaining moisture within folded tissues. In subtidal zones, they expand fully, maximizing capture surface. This plasticity allows them to endure fluctuating conditions without locomotion. Adaptation occurs not through relocation, but through modulation of form. The final revelation is that the sea anemone embodies a model of predation divorced from speed or spectacle. It does not pursue, chase, or display dominance. Instead, it defines territory chemically. The water column becomes its hunting ground, not through motion, but through reach. Any organism that brushes too near enters a zone of microscopic harpoons and neurotoxic peptides. The anemone survives because it transforms passivity into potency. In the reef's intricate architecture, where color and motion compete for attention, the sea anemone stands quietly, tentacles drifting like petals in current. To the untrained eye, it is decoration. To plankton and unwary fish, it is perimeter and trap. Within its tissues, algae converts sunlight to sustenance. Within its tentacles, venom waits coiled in capsules smaller than grains of sand. The ocean reveals once more that danger does not always advance. Sometimes it blooms, rooted and radiant, a living contradiction in which softness conceals strike and stillness conceals power. The sand erupts without warning. A foot presses down into what seemed like nothing more than warm seabed, and in the same fraction of a second, a whip-like tail snaps upward with surgical speed. There is no roar, no charge across distance. Only pressure, reflex, and a serrated spine driving upward into flesh. Venom follows along grooves carved into the barb, flooding the wound with compounds designed to amplify pain and enforce memory. The stingray never chased the intruder. It lay still, invisible beneath a veneer of sand, and when the world bore down on it, the seafloor itself struck back. Stingrays are masters of concealment, not through pigmentation alone, but through physics. Their bodies are flattened into broad discs. Pectoral fins fuse to the head in a continuous plane that distributes weight evenly across soft sediment. By undulating these fins in smooth waves, they glide just above the substrate or settle gently into it, stirring minimal turbulence. Once at rest, they fan sand across their backs with subtle motions, covering their dorsal surface while leaving spiracles exposed to draw water into the gills. What appears to be barren seabed may conceal a living animal beneath a thin layer of grains. This is camouflage born from hydrodynamics and morphology combined. Unlike venomous predators that deploy toxins as hunting tools, stingrays carry their chemical arsenal as contingency. The tail, often slender and whip-like, bears one or more barbed spines composed of dentin-like material sheathed in integument that contains venom glands. When the spine penetrates tissue, the sheath tears, releasing venom into the wound. The barb's serrations can anchor in flesh, increasing tissue damage upon withdrawal and ensuring that the deterrent effect is not subtle. The venom contains proteins and peptides that trigger intense nociception and inflammation, producing pain disproportionate to the size of the wound. This is emergency deterrence in its most visceral form. The narrative reversal lies in the distinction between capability and character. Stingrays are not aggressive chemical hunters. They feed primarily on invertebrates buried within sediment, using electroreception to detect faint bioelectric signals emitted by prey muscles and nerves. Specialized organs known as ampullae of Lorenzini allow them to sense voltage gradients in the water, pinpointing hidden mollusks and crustaceans beneath sand. Once detected, the ray employs suction and jaw pressure to excavate and crush prey. Venom plays no role in this process. The stingray's identity is not defined by its toxin, but by its sensory acuity and benthic lifestyle. This separation of feeding and defense reveals an elegant allocation of biological resources. Venom production and maintenance carry metabolic cost. By reserving toxin for last resort situations, stingrays avoid unnecessary expenditure while preserving a powerful deterrent. The spine remains passive until threatened, transforming the tail into a sudden arc of consequence only when escape routes are compromised. In this way, the ray balances vulnerability and power, relying first on camouflage and flight, and only finally on chemical retaliation. The seafloor habitat intensifies this strategy. Coastal shallows and estuaries often teem with larger predators such as sharks, dolphins, and crocodilians. For a bottom-dwelling animal, immediate vertical escape may not be feasible. Instead, concealment reduces encounter rates. When concealment fails, the tail strike enforces a boundary. Predators that attempt to seize a ray risk injury severe enough to alter future behavior. Over time, this feedback loop conditions caution among potential attackers. The stingray's venom shapes predator perception, contributing to the behavioral landscape of the ecosystem. There is also structural compromise embedded in the spine itself. Unlike the continuously replaced teeth of sharks, stingray spines are shed and regrown periodically. Damage or loss leaves the animal temporarily less defended. During these intervals, reliance on camouflage and early detection becomes paramount. The venomous barb is not permanent armor, but renewable weaponry, subject to cycles of vulnerability and restoration. Evolution has favored a system that balances risk and regeneration. Reproduction adds another dimension. Many stingrays are viviparous, nourishing embryos internally before giving birth to fully formed young. In nutrient-rich coastal nurseries, juvenile rays must navigate predator-dense waters with limited size advantage. Their small tails already bear functional spines, granting them defensive capacity from early life stages. This inheritance of chemical protection underscores the importance of venom not as ornament, but as survival baseline in high-risk habitats. Yet stingrays themselves are not immune to predation. Large sharks and orcas have been observed flipping rays to expose their softer undersides, circumventing the tail's reach. This tactic illustrates the ongoing evolutionary chess match between defense and counter-defense. The stingray's spine deters many threats, but specialized predators adapt behaviorally to neutralize it. Chemical defense is powerful but not absolute. Environmental change introduces further complexity. Coastal development and sediment disruption can alter benthic substrates, affecting the ray's ability to bury effectively. Pollution and habitat degradation may reduce prey availability, forcing rays into unfamiliar areas and increasing human encounters. The stingray's defensive reflex, evolved in response to natural predators, becomes hazardous when triggered by inadvertent human contact. The pain inflicted is not malicious intent, but evolutionary reflex intersecting with altered ecosystems. Within the broader marine narrative, stingrays exemplify a strategy rooted in restraint. They glide with grace, wings rippling as though in slow flight over sand plains. Their eyes protrude slightly above the body, scanning for threat, while spiracles pulse rhythmically to draw water across gills. Nothing about their posture suggests aggression. Yet coiled within the tail is potential energy, ready to convert compression into retaliation. This duality defines their place in the ocean. The ecological twist emerges in recognizing that their venom, though potent, is secondary to their true mastery, sensing and shaping the benthic environment. By feeding on burrowing invertebrates, stingrays aerate sediment and influence invertebrate populations, indirectly affecting nutrient cycling. Their foraging pits alter microhabitats, creating depressions that can become refuges for smaller organisms. The ray is not merely occupant of the seabed, but engineer of its texture. In the shifting light of shallow waters, a stingray lifts gently from the sand, revealing the pale imprint where it once lay concealed. It glides forward with unhurried rhythm, tail trailing like a signature written in water. The spine remains still, an unsheathed warning awaiting necessity. The ocean's lesson here is subtle but profound. That toxicity need not define identity to shape survival. The stingray survives not because it seeks confrontation, but because it carries within its elegance a final argument against it. When pressed, it answers with pain sharp enough to etch memory into nerve and mind alike. When left undisturbed, it resumes its quiet patrol across the sea floor, guided by electric whispers beneath the sand. In this balance between grace and defense, the stingray reveals a deeper truth about marine life. The most dangerous weapon is often the one that is never displayed, integrated seamlessly into a body built for calm. waiting for the rare moment when the world steps too close and learns that even the gentlest glide can conceal a barbed reply. The coral screams in silence. Beneath a halo of drifting plankton and refracted light, a living reef polyp retracts as a pale, many-armed form settles over it like a slow eclipse. Tube feet grip the limestone skeleton with hydraulic precision, and then the stomach emerges from the starfish's central disc, everting outward in a pale membrane that spreads across living tissue. Enzymes flood the coral's surface, digesting it externally before absorption. Within hours, vibrant polyps become exposed white skeleton. The predator does not bite. It dissolves. The crown-of-thorns starfish is not subtle in appearance. Its body radiates outward in multiple arms, each one armored with long, needle-like spines coated in venom. These spines are not mere deterrents. They are structural investments in survival. When pressed, broken, or mishandled, they penetrate skin easily and introduce venom that induces pain, swelling, and inflammatory responses. For potential predators such as reef fish or curious mammals, contact is costly. The starfish carries its chemical defense outward in every direction, transforming its entire body into a bristling perimeter. It does not need to chase enemies away. It ensures that contact is remembered. Yet the venom is only the first layer of influence. The ecological power of this animal lies in its appetite. Crown-of-thorns starfish specialize in consuming live coral tissue, particularly fast-growing branching species. They extend their stomachs over coral surfaces, secreting digestive enzymes that liquefy living cells while leaving the calcium carbonate skeleton intact. This feeding strategy allows them to process large areas efficiently without the need for internal mastication. Over time, a single individual can clear significant patches of reef. When populations remain balanced, this grazing can contribute to coral diversity by preventing dominance of one species. But when numbers surge, the dynamic shifts dramatically. The narrative reversal emerges in the scale of impact. An animal that appears slow and limited to the seafloor becomes a force capable of reshaping entire reef systems. Population outbreaks, sometimes fueled by increased nutrient runoff that enhances larval survival, can result in vast swaths of coral consumed. Without live coral, the structural complexity of the reef diminishes, affecting fish that rely on branching forms for shelter and breeding. The predator of coral becomes, indirectly, a catalyst for trophic cascades. Fish populations decline, algal growth may increase, and the reef's three-dimensional architecture erodes. A chemically defended invertebrate alters the future of a habitat. Understanding this dynamic requires examining life history. Crown-of-thorns starfish release vast numbers of gametes into the water column during spawning events, relying on external fertilization. The resulting larvae drift as plankton before settling onto suitable substrates. In nutrient-enriched waters, larval survival rates can increase significantly, producing recruitment pulses that swell adult populations. Human activities such as agricultural runoff amplify this effect, linking terrestrial nutrient cycles to marine outbreaks. The starfish's ecological power is thus intertwined with watershed management and coastal development. Chemistry and ecology intersect across ecosystems. Predation on crown-of-thorns starfish is constrained not only by spines, but by chemical deterrents. Certain predators, including some species of pufferfish, triggerfish, and the giant triton snail, have evolved strategies to consume them despite venomous defenses. The triton snail in particular can subdue the starfish by secreting immobilizing saliva and avoiding the densest spines. These interactions highlight evolutionary negotiation. The starfish's spines reduce predation pressure, enabling population growth when predators are scarce. Conversely, overharvesting of natural predators can remove checks on starfish numbers, allowing outbreaks to intensify. The reef's balance hinges on these relationships. The crown-of-thorns starfish also exhibits behavioral adaptations that maximize feeding efficiency. It often feeds at night, reducing exposure to visually oriented predators and minimizing detection. During daylight hours, individuals may retreat into crevices, their spines protruding defensively from rock cavities. This dial rhythm conserves energy and reduces risk. Movement is achieved through coordinated tube feet operating via a water vascular system, allowing steady progression across coral surfaces. The starfish's locomotion may appear slow, but in ecological time, it is relentless. Venom in this context serves not to secure prey, but to protect the grazer. Coral polyps lack the mobility to escape predation, but the starfish must withstand attention from fish and other reef inhabitants while feeding. Spines provide a chemical buffer, granting uninterrupted access to living coral. This synergy between defense and feeding amplifies impact. The starfish does not merely survive on the reef, it feeds with reduced interference, accelerating habitat modification during outbreaks. The ecological twist deepens when considering coral resilience. Healthy reefs can recover from limited grazing, as surviving polyps reproduce and regrow over skeleton. However, when stressors such as rising sea temperatures and ocean acidification weaken coral physiology, recovery slows. Crown-of-thorns outbreaks superimposed on climate stress can tip reefs past thresholds of resilience. In this interplay, the starfish becomes an amplifier of vulnerability. Its chemical armor and feeding capacity intersect with global environmental pressures, producing outcomes that reverberate beyond individual reefs. Yet the starfish itself is subject to environmental variability. Larval stages are sensitive to temperature and water chemistry, and extreme conditions can reduce survival. Disease outbreaks among starfish populations can also collapse numbers abruptly. These fluctuations remind us that no ecological force operates unchecked indefinitely. The crown-of-thorns starfish is both driver and participant in dynamic systems. As the feeding front advances across coral colonies, white scars mark its passage like ghostly footprints. Fish that once darted among branching structures find fewer refuges. The complexity that supports biodiversity thins. Yet in moderation, the starfish's grazing may promote coral diversity by preventing competitive exclusion. It is not inherently villainous. It is a regulator that becomes destructive when regulatory mechanisms fail. The final revelation is that venom here is not merely a personal defense, but an enabler of ecosystem engineering. The spines discourage predation, allowing sustained feeding that can reshape habitat. The starfish's chemical shield protects an appetite capable of altering reef architecture. It is both toxic organism and agent of transformation. In the dim blue of a reef at dusk, a crown-of-thorns starfish spreads across coral like a many-armed constellation fallen to earth. Each spine glints faintly, a reminder of pain encoded in tissue. Beneath its body, coral life dissolves into nutrient flow. The ocean reveals a paradox, that beauty and destruction can coexist in one organism, that a creature armored in venom can influence not just its survival, but the destiny of entire ecosystems. The crown-of-thorns starfish stands as testament to the power of chemistry, not only to defend a body, but to redefine a landscape. A shadow passes over the reef and the mouth opens anyway. Teeth descend toward what appears to be a simple, bristling sphere wedged between coral heads, and in the next instant the bite collapses into regret. Spines snap and penetrate soft tissue, some breaking off under pressure, others flexing without yielding. Tiny, grasping structures clamp onto the intruding surface, increasing contact area and driving irritation deeper. The sea urchin does not recoil. It absorbs the impact and answers it with geometry. What looked like an exposed weakness is in fact a fortress built outward in every direction. The sea urchin embodies one of the ocean's most ancient architectural solutions, radial symmetry, armored in calcite. Its body is encased in a rigid test composed of interlocking plates, a skeletal sphere assembled from mineral crystals precipitated within living tissue. From this shell radiate spines that vary in length, thickness, and function across species. In some urchins, the spines are primarily mechanical deterrents, sharp and brittle enough to lodge in flesh. In others, the spines are accompanied by venom glands or by pedicellariae, minute pincer-like appendages capable of gripping and injecting irritants. The surface becomes a living minefield, each spine and pincer a localized warning. The power of this system lies in its integration of chemistry and structure. An urchin cannot sprint across sand to evade a predator. Its tube feet move in coordinated waves driven by hydraulic pressure within a water vascular system, granting slow but deliberate locomotion. Its survival depends not on speed, but on discouragement. By presenting a spherical array of spines that complicate grasping from any angle, the urchin transforms its immobility into defense. A predator that attempts to bite must negotiate a forest of needles first. If penetration occurs, toxins may amplify the pain, reinforcing avoidance behavior in future encounters. The urchin invests in memory within its enemies. The narrative reversal emerges when one considers that beneath this spiny exterior lies a mouth. On the underside of the sphere sits Aristotle's lantern, a complex arrangement of 5 calcified teeth that operate in coordinated motion to scrape algae and biofilm from rock surfaces. The same animal that appears as a passive obstacle is an active grazer, capable of reshaping benthic landscapes. Its feeding pressure can regulate algal growth, maintaining open substrate for coral recruitment. In balanced ecosystems, urchins contribute to reef resilience by preventing algae from smothering corals. The fortress grazes. This grazing capacity becomes transformative when predator populations shift. In regions where urchin predators decline due to overfishing or disease, urchin numbers can surge. Increased grazing may strip algae so thoroughly that barren grounds emerge, expanses of rock scraped clean of vegetation. While such areas may seem devoid of life, they represent a state driven by unchecked herbivory. The urchin's chemical and mechanical defenses enable persistence at densities that alter ecosystem structure. Its survival strategy scales upward into ecological influence. Venomous species intensify this dynamic. Some urchins possess spines coated in toxins that induce burning pain, swelling, or muscle cramps upon contact. Others rely on pedicellariae that clamp down and inject irritants when stimulated. These microstructures function independently of the main spines, responding to touch with reflexive closure. The predator's skin becomes a testing ground for chemical negotiation. Even if a spine does not pierce deeply, the combined irritation from multiple contact points can deter continued attack. The urchin's defense is distributed rather than centralized. Energy economics shape this arrangement. Maintaining a rigid mineral test and constantly regenerating broken spines demands metabolic investment. Calcium carbonate deposition requires access to dissolved ions and energy derived from feeding. The urchin balances the cost of armor with the benefit of survival in predator-rich habitats. Spines broken during encounters regrow over time, restoring the defensive field. The body plan is both durable and renewable, reflecting millions of years of selective refinement. The ecological twist deepens in the interplay between urchins and coral reefs. Healthy predator populations keep urchin numbers in check, preventing overgrazing. When predators decline, urchin populations may explode, shifting reefs toward barren states. Conversely, in systems overwhelmed by algae due to nutrient enrichment, urchins may serve as crucial regulators, restoring balance through intense grazing. Their impact is context-dependent, oscillating between guardian and disruptor. The same spiny persistence that deters a fish can sculpt entire habitats. Sea urchins also participate in symbiotic relationships. Certain fish and crustaceans seek refuge among their spines, exploiting the deterrent field as protection against predators. Juvenile fish may hover within the spine canopy, shielded from larger threats. In this way, the urchin's defense extends beyond itself, creating micro-habitats of safety. The fortress becomes sanctuary. What first appeared as solitary stubbornness reveals itself as node within a broader network of associations. Reproduction introduces yet another dimension. Many urchins release gametes into the water column, relying on synchronized spawning to maximize fertilization success. The resulting larvae drift as plankton before settling and metamorphosing into radial juveniles. Ocean currents carry genetic material across distances, linking distant populations. The armored grazer, anchored to rock, participates in oceanic dispersal far beyond its limited adult movement. Persistence at one site does not imply genetic isolation. Predators that specialize in consuming urchins evolve counter-strategies. Some fish approach from angles that minimize spine contact, biting selectively at exposed regions. Sea otters in certain ecosystems use tools to crack open urchin tests, bypassing spines entirely. These adaptations illustrate the ongoing evolutionary dialogue between defense and innovation. The urchin's armor provokes ingenuity. As currents sweep across rocky substrates, urchins adjust orientation subtly, aligning spines with flow to reduce drag and maintain position. Tube feet extend and retract in coordinated patterns, allowing gradual repositioning toward food sources or shelter. Though movement is slow, it is purposeful. The radial symmetry that seems archaic proves efficient for distributing mechanical stress and maximizing defensive coverage. The final revelation is that the sea urchin's apparent simplicity masks profound ecological consequence. Its spines deter mouths and feet alike, turning each attempted bite into a calculated risk. Its grazing can either preserve coral by restraining algae or strip landscapes bare when unchecked. Its venom and pedicelaria amplify the cost of intrusion, embedding caution within predator communities. In the shifting mosaic of reef and rock, the urchin endures. It does not chase, threaten, or advertise. It remains, bristling quietly, test gleaming beneath spines that catch the light. Beneath that armor, teeth scrape steadily at stone, converting algae into energy and influencing the future of the reef. The ocean teaches again that survival need not be swift or dramatic. Sometimes it is stubborn, radial, and resolute, an animal that wins by making every approach a negotiation, weighted against the attacker. In the quiet persistence of the sea urchin lies a lesson in endurance. That permanence, reinforced by chemistry and geometry, can be as transformative as any predator. A pulse of agony detonates beneath the surface before the water even ripples, a biological ambush so fast that the nervous system registers catastrophe before the mind identifies the attacker. In less time than it takes for a human reflex arc to complete its loop from skin to spinal cord and back again, thousands of microscopic harpoons have already fired. Each one is a coiled biochemical spring, compressed within a capsule no wider than a grain of dust, triggered by the faintest mechanical brush against living tissue. The box jellyfish does not bite, does not chase with jaws or claws. It converts contact itself into a weaponized event, transforming the ocean into a minefield where touch becomes trauma. This is predation stripped to its chemical essence. No roar, no shadow, only a transparent geometry drifting in warm coastal currents, its cube-shaped bell refracting light so subtly that it vanishes against the glitter of the tropics. What appears delicate is in fact an arsenal. Along each tentacle, thousands upon thousands of nematocysts lie primed. Each one a single-use projectile driven by osmotic pressure that can accelerate its filament at speeds among the fastest cellular motions in the animal kingdom. The discharge is not random. It is mechanically gated, activated when the cilia-like triggers sense deformation consistent with living prey rather than inert debris. The venom delivered is not a simple irritant, but a complex cocktail of pore-forming toxins and cardiotoxic compounds that disrupt ion channels in cell membranes, allowing calcium and sodium to flood tissues that depend on precise electrical gradients. Pain is the first signal, but it is merely a symptom of a deeper assault on neuromuscular coordination and cardiac rhythm. In severe exposures, the toxins compromise the heart's ability to maintain synchronized contraction, creating arrhythmias that can escalate into systemic collapse. The box jellyfish does not overwhelm by mass. It overwhelms by biochemical precision. Yet the narrative reversal lies in what this weapon is truly designed for. The box jellyfish is not evolved to hunt humans, nor to terrorize swimmers. Its venom is optimized for small, agile prey that would otherwise outmaneuver a gelatinous body in open water. Unlike many other jellyfish that drift passively, box jellyfish possess a surprising degree of directional control. Their bell pulses rhythmically, propelling them at speeds sufficient to pursue fish larvae and small schooling species through mangrove channels and estuarine shallows. Embedded along the bell are clusters of simple but functional eyes, capable of detecting light gradients and obstacles. They navigate towards shaded routes where juvenile fish congregate, steering with a level of sensory integration that challenges the stereotype of canidarians as mindless drifters. The venom that incapacitates a human is a scaled-up effect of a tool evolved to immobilize prey within seconds, preventing escape in an environment where hesitation equals starvation. The ecological stage for this predator is not the abyss, but the sunlit littoral zone, where tides concentrate nutrients and juvenile fish shelter among submerged vegetation. Here, energy flows rapidly through food webs, and metabolic rates run high in warm water. For a gelatinous predator with limited structural support, efficiency is survival. The box jellyfish cannot afford prolonged chases. Its mesoglia-filled body lacks the musculature for sustained pursuit. Instead, it relies on ambush facilitated by transparency. Its tissues are nearly invisible because they lack dense pigments and scattering structures, allowing light to pass through with minimal reflection. In tropical waters already saturated with glare and particulate shimmer, the animal becomes an optical ghost. Prey do not see a silhouette. They encounter an interface. And when they do, the tentacles wrap reflexively. The nematocysts fire in synchronized bursts, and paralysis follows almost instantly. The venom's potency also serves a defensive function in an ecosystem crowded with predators. Sea turtles, certain fish, and specialized nudibranchs have evolved partial resistance to jellyfish toxins, feeding on tentacles without succumbing to the full biochemical cascade. This is the arms race that underlies marine trophic dynamics. Toxins escalate, resistances evolve, and ecological niches shift accordingly. The box jellyfish sits within this web, not as an isolated terror, but as a node in a network of selective pressures. Its venom shapes the behavior of fish schools, influences the distribution of juvenile cohorts, and indirectly alters nutrient pathways by modulating which prey survive to maturity. A single sting can reverberate through a microhabitat, removing an individual from a cohort and subtly adjusting competition among survivors. Chemical predation becomes a force sculpting community structure. The true twist emerges when we consider that the box jellyfish's greatest strength is inseparable from its vulnerability. Its body is composed largely of water, its tissues fragile and easily torn by turbulence. Storm surges and strong currents can shred tentacles, dispersing fragments that still retain functional nematocysts. Detached tentacles continue to sting, extending the animal's chemical footprint beyond its physical integrity. Yet this fragility limits its distribution. It thrives in relatively calm coastal zones, where wave energy is moderated by reefs and mangroves. The very habitats that protect its structure also concentrate human activity. Thus, encounters are not the result of aggression, but of overlapping ecological preferences. The jellyfish seeks nurseries rich in prey. Humans seek warm shallows for recreation. Two species drawn by productivity meet at the interface of survival and leisure. Within its simple nerve net lies another paradox. Without a centralized brain, the box jellyfish coordinates complex behaviors through distributed neural circuits. Sensory inputs from its eyes and statocysts integrate to guide movement, demonstrating that sophisticated responses can emerge from decentralized systems. This challenges assumptions about the hierarchy of nervous organization. Intelligence, in this context, is not a matter of cortex or cognition, but of adaptive patterning. The jellyfish does not deliberate. It responds with evolutionary refinement encoded in cellular machinery. The harpoon fires because molecular tension has reached a threshold. The bell contracts because ionic gradients shift across membranes. Life operates through chemistry long before consciousness interprets it. As the sun descends and the water cools slightly, box jellyfish may alter their vertical position in the column, tracking prey movements that follow diel cycles. Fish larvae often rise towards surface plankton under fading light, and the jellyfish adjusts accordingly, exploiting predictable behavioral rhythms. This temporal synchronization exemplifies an ecological alignment where predator and prey are locked in mutual influence. The venom's speed ensures that even brief contact is sufficient. Prolonged entanglement is unnecessary. Energy is conserved, metabolic expenditure minimized. In a realm where caloric intake must balance buoyancy maintenance and reproductive investment, efficiency becomes evolutionary currency. Reproduction introduces yet another layer to the story. After a period in the planktonic medusa form, box jellyfish trace their lineage back to a benthic polyp stage, anchored to substrates and reproducing asexually before metamorphosing into free-swimming forms. This life cycle bridges sessile and mobile existence, allowing populations to persist through environmental variability. Polyps can endure conditions that would challenge delicate medusae, serving as reservoirs that reseed coastal waters when temperatures and prey densities align. The predator we fear as a drifting apparition is, at another stage, a microscopic adherent clinging to rock, awaiting transformation. Survival is not a single strategy, but a sequence of forms adapted to shifting pressures. The ocean's chemistry underpins every facet of this organism's power. Salinity influences nematocyst discharge dynamics. Temperature modulates metabolic rate and venom efficacy. Climate-driven changes in coastal conditions may alter bloom frequencies, shifting the spatial and temporal patterns of box jellyfish presence. Increased nutrient runoff can elevate plankton abundance, indirectly boosting prey populations and supporting larger jellyfish cohorts. Thus, the sting felt by a swimmer is entangled with global processes, from watershed management to ocean warming. The animal becomes a bio-indicator of coastal productivity and imbalance, its abundance reflecting the state of marine ecosystems. In the final analysis, the box jellyfish embodies a form of predation that defies the archetype of muscular dominance. It is neither armored nor toothed, neither swift in the mammalian sense, nor endowed with complex cognition. Its supremacy lies in microscopic architecture and biochemical choreography. Each tentacle is a filament of evolutionary trial. Each nematocyst a perfected microdevice forged through millions of selective encounters. What seems fragile is fortified by chemistry. What seems passive is strategically positioned within energy-rich corridors of the sea. And so the ocean reveals another of its reversals. The creature that appears as a drifting ornament is, in truth, a convergence of physics, physiology, and ecological timing. Its weapon is not brute force, but instantaneous transformation, the conversion of touch into systemic shock. In the silent warmth of tropical shallows, where light fractures into wavering patterns and juvenile fish dart between roots, the box jellyfish moves with unassuming grace. There is no warning display, no audible threat. Only a transparent cube pulsing through water, trailing invisible lines of consequence, reminding us that in the marine world, the most devastating predators may be those that wage war at the scale of molecules, turning the very chemistry of life into a blade too small to see and too fast to escape.